Okay, great. So good morning, everybody. I'm going to be talking about stochasticity in epithelial tissues. I think some of you don't need the introduction to epithelial tissues, but I'm still going to give you a little bit of what epithelial tissues are and why do you need to even look at the stochasticity in these systems. So this is an example of a top view of an epithelial monolayer. Um, and sorry, I can't go forward. I'll go it from, okay, we'll start again. So look at this tissue from the top and you see a lot of dynamics. So you see a lot of activity going on and cells are trying to maintain this homeostatic layer over the adult life by maintaining a balance between cell death and cell division. And then the home density remains more or less constant. So here what you also notice is if you are a physicist, you would notice that these cells are not only biologically active, but they are also resembling somehow soft matter. Because they are soft, they are deformable. So that means that they get deformed upon application of force from their neighbors. So if the neighbors are applying a pushing or a pulling force, they would deform, they will respond to that force. So they are both a combination of both soft and active matter in this case. And actually, as a physicist, you can also put this in the perspective of dynamic heterogeneity. So, it's been described, these epithelial tissues were described as demonstrating these traits of dynamic heterogeneity. Now, for biologists, I will explain what that means. So, if you just simply put these colloidal particles or any particles which are non-equilibrium in nature in a low density in a constrained environment, they will diffuse. So that means that they will move about in space and they are happily migrating in that constrained space if there is space available. But when you increase the density of these particles, what they are going to achieve is some kind of a jamming configuration, which you I think also heard in Tapo's talk a little bit in the previous talk. So they attain this jamming conformation and they increase their cooperativity, meaning that the neighbor can only move when the neighboring particle will cooperate with the movement. So that means that if a lot of these particles are crowded together, they can only move when they cooperate with each other. The cooperativity is increasing and their overall movement is reducing. And so similar kind of characteristics. So this is a very typical soft matter, non-equilibrium matter characteristics that sand, grains, coffee beans, or colloidal particles display. Now you can also see or view epithelial systems or epithelial tissues, epithelial cells in a similar way, where when you increase their density, they're going to achieve this kind of jamming configuration. Their movement overall is reducing. So that means that when they're at a lower density, similar to these particles, there's a lot more movement, which is very random and dispersed, but higher. But when they increase their density, their movement is going to reduce. Overall movement is going to be low and low as you increase the density. But the movement is becoming more and more cooperative. So you can see, I don't know if you can appreciate it, but the arrows are more correlated with their neighbors. So that means that cooperativity is increasing, movement overall is reducing, and that's a similar property of non-equilibrium matter that you see in nature. So this paper also actually shows this plot where when you enhance or you increase the cell density, the number of cells that cooperate with their neighbors gradually increases over increase in cell movement. So what does that mean? Now you can also look at do an experiment with the epithelial tissue. So as I'm an experimentalist and I use these concepts to understand biology and of course concepts of physics and bioengineering experiments to do all sorts of and understand the biological systems and their mechanics. So what we did was you do an experiment, and the experiment that we did here was traction force microscopy. I don't know how many of you have heard about traction force microscopy. Okay, some of you, or many of you. Not many of you have heard still, so I'm going to tell you what it is. It's very simple. So you take a gel, hydrogel, just a gel, and you put some fluorescent markers, some spherical beads in these hydrogel, and you put your cells on top of this hydrogel. Now, when the cell is doing any movement or it's dividing or any activity it's doing, it's going to displace these beads that are underneath the cell. So from the displacement of the beads, you can calculate how much force the cell is exerting on its substrate. That is called a traction force. So it's similar to a frictional force where your car is driving on the road, how much force it's exerting on the road. So and then if your epithelial tissue is a homeostatic monolayer, you can assume that the cell substrate force is somehow balanced by the cell force, and you can infer the monolayer stress from this kind of calculation. So when you do that, you're going to get this kind of stress profile, which is very interesting in its way, because it shows this stochasticity in the system. So it shows peaks and valleys of high and low stresses. That means that there are cells that cooperate forces with their neighbors. So I'm a cell, I'm pulling, and this force is being transduced to my neighbors up to a certain distance, and then it's going to decay. And then again, there's a peak. So there's these peaks and valleys that shows cooperativity in the epithelial systems, and also stochasticity, that not all the cells have the similar level of forces in the tissue. So that this is all very nice. You see a stochasticity in their physical forces that they exert on their neighbors and on the substrate. But what does it mean? How can you think about it as a function? And would it impact any kind of function of that? Yes.
You mean the range of forces is stochastic, or whether it extrudes a force on its neighbor is stochastic? Can you explain that part?
So what I mean is that if you can you see cells underneath this stress landscape? So this cell, so the cells here are exerting much less forces than the cells here, for example. And it's stochastic, meaning that it's heterogeneous in time as well. So if you view it over time, basically what I want to say in short is that this high force is not tied to a cell. It's a dynamic heterogeneity landscape which will change over time. So if you give it time, this peak will actually shift to another location. So it's not tied to individual cell, but it's tied to the property of the tissue as it is. So basically the overall collective dynamics of the cells. You got it? Okay. So one can ask a question, what does it imply for the tissue and for its function? So what we did was, you can actually look at several functions of the tissue at this point. You can look at how the stochasticity will change the way the tissue will, take the metabolites, and when you eat the food, your metabolites are going to, your cell epithelial tissues are going to do this nutrient exchange from the tissue and get the homeostasis going. So how does it how does it impact that function? Or you can look at the cancer initiation and progression, or you can look at wound healing. So there's several functions that epithelial tissues or properties that epithelial tissues display, and one can look at several functions on how this dynamic heterogeneity is impacting one of these functions. So in this study, we actually chose to look at the wound closure, which is the tissue remodeling part of the wound closure, meaning that at the end of the wound, when the cells collectively move to heal the gap of the monolayer. So that means that if your monolayer is looking like this and there's a sudden wound, the cell will then transition from these jammed tissue to an unjammed system, where the cells then will collectively migrate to heal the wound. So in this migration, there's interesting things that you observe, and one of these things is this leader cell formation at the edge, at the tip of this, at the edge of this wound. So do you see these cells that are forming these very large protrusions? Those are the leader cells that are leading the whole collective forward. Yes, you have a question?
How you create this experiment like?
So this is an in vitro wound healing system. Actually, it's very easy. You just throw some cells on a dish or on a gel, in whichever substrate you want, and let it reach this homeostasis environment. And then you take a tip and you scratch.
Wound, or like how you differentiate in experiments between a wound and normal epithelial cell, like?
So wound is where you have a wound, so basically a scratch. So it's a scratch wound assay where a homeostatic monolayer, we just scratch it with the tip, and there will be a gap that will be generated similar to a wound. So if you have a wound in your hand, there will be a gap between your epithelial cells are layering the top of your tissue, right? So there will be a gap that will be formed upon a wound, and that gap needs to be filled. So the cells will then recognize that gap and start to move. And now this kind of assay you can also do in in vitro without a scratch. So basically it's called confinement release assay, I think, where you just confine the cells in a space and then remove the confinement and allow the cells to migrate. So that is a little bit different than this, because here you are creating an actual wound But in both cases, we see these leader cell formation and the collective motion of the cells.
Ma'am, I have a doubt. So here, the number density is constant at this time scale, or like
Oh, sorry.
the online participant? Sorry, should have made it clear.
I'm like, why don't I see her? Can you say that again, the question?
Here, the number density, like, is it like maintained constant, or like what is happening with the number? Like, are they
The
generating?
number of the cells. So you mean whether they're dividing or not? That's your question, right?
Yeah, so if they're dividing, then the number density is
So
not.
yeah, so the thing is, this is a confluent monolayer, and when we scratch, so actually the cell division is very minimum at this stage because the monolayer has reached the homeostasis. So when they reach, only when they reach the homeostasis and the collective dynamics or the dynamic heterogeneity of those properties, only then we scratch them. I will show you. Actually, this is a very good question. So I'm going to show you an example where we do not let them reach the homeostasis and we scratch them earlier, because we wanted to see what might be the role of cell division, or if they're not able to maintain this property of pulling each other with these cooperative forces, then what happens, how does it impact on the wound? That I'm going to show you in a few slides. But here the cell division is very minimum. You see some division happening here, but it's not so the most of the energy that the cells are exerting is on the migration, because they see this gap and they want to fill the gap as soon as possible. So they're just migrating towards the gap. You have another question?
Are the boundary cells somehow more illuminated, like through some special means, like on the left-hand side picture?
This one?
Yeah.
So this is just a phase contrast microscopy. So basically when you scratch, because it's a white light phase contrast microscopy, there's a different contrast because they are damaged cells. I think that's what you see. They're not illuminated differently. It's the same amount of light. You have a question?
What cell lines you are using?
Sorry, lens?
What cell line?
Cell line. So this is an MDCK cell line.
Second, first one I can see it's a phase contrast image, and what about the second?
This one is the life actin. I should have written it somewhere. It's not. Okay, sorry. It's an actin labeling. Basically, the cells which are expressing live actin, GFP actin.
And the time scale.
Time scale is not. So it's very. I think it's about 10 hours. Yeah, 10 hours.
Okay. Thank you.
So I don't know what a phase contrast microscope is. Can you please explain what that is?
Sure. So a phase contrast microscopy is a standard microscopy that biologists use all the time to view cells. So it's just your white light is coming, and then you get a contrast from the phase ring that is there on the objective. It's a bright field microscopy. It's not dark field microscopy. It's a bright field microscopy, but it has just that the objective has a phase ring to get this contrast that you see. So it's actually exactly like the bright field microscopy, just a phase ring that is there on the objective to get a little bit more contrast to see the cell boundaries. Yeah. So.
Yeah. Do you know which of the cells will become leaders and which of them?
Yeah, so that is what I'm going to show you actually. So this is a very good question. So if you just do this assay, actually I have it in the next slide. So, if you just do a confinement release assay, you will see that all of the cells look exactly the same, but then suddenly some of the cells will become leaders and the others will not. So the question is, how do the cells choose between who will become the leader? And that is where I was coming at when I'm talking about the dynamic heterogeneity of the monolayer. So this is actually this movie will answer your question, the one that I'm going to just play now, the one that you see in colors. So this is actually a monolayer stress microscopy over time, and I overlaid it with phase contrast movie, and this in the pink you see accumulation of higher stress. So this is the scale bar. You can see that the pink is where you see much higher stresses. And if I play this movie, it's kind of self-explanatory, I think. I'm going to, if it plays at all. Okay, now, please. So you see the regions that you see much higher accumulation of stresses, that's where you see the formation of leaders. So where I'm getting at is that the formation of leader cells during wound healing is not a cell autonomous behavior, but it's more of a collective behavior of the tissue itself, or the stochasticity that I was just mentioning you, where you see cooperation of higher forces within the monolayer that will determine where the leaders will be formed when there will be a wound in the future. And one can also do an experiment to actually prove that. So this was a monolayer stress microscopy where the one that I was just showing you, where we did a traction force microscopy and then inferred cell-cell stresses, and we see accumulation of high stress in the regions where the leader cells are going to form. But as a bioengineer, we want to demonstrate everything also in the lab, that this is what is really happening. So you can actually try to pull. So what I'm going to do now, if you have this trench, and you have a cell here and a cell here, and I pull from the back, the cell that is in the front will actually form the leader cell. So that is the hypothesis, right? So that's where you see the accumulation of forces. And what I'm trying to say, the hypothesis is that where you see higher forces, that's where you're going to see the leader cells in the future. So we did this experiment. So this is a PDMS membrane. And we placed cells here in a confined space and made a trench, something like this, a trench, and then let the cells migrate across the trench. And when we see one layer of cells ahead of the trench, we pull. So we pull this PDMS membrane from the back. So that means we are just pulling this. I can't pull this, it's too heavy. but you pull this from the back, and that means that the yellow cell is going to pull on the pink cell. And when you do this, it's very nice demonstration that you can see. So this is a trench, and we pull from the back, and you see these leaders forming in the front. And then they lose their leadership. This is because there's an existing leader already here, and there's a territory of leadership, which is also very interesting. It's an interesting concept. So I will not talk about too much about everything, but just giving you a glimpse of what you can understand from it. So basically, there's a leader cell here and a leader cell here, and you force formation of protrusions or leader cells behind or ahead of this trench, and then over the time, these leaders are turning back. So pulling forces from the bulk allow protrusion formation in the future leaders. Somebody has a question here.
One thing, like when we are talking about cell migration, so like if the cells has to fill some space, so shouldn't be there like generation of some cells at some other place, or is it like the density of the cells is changing?
Yeah, so there will be cell division also. When you mean that when the cells are filling the gap, there will be also division in the back of the cell. So the thing is, what is happening? So this has been already actually shown in some of the studies, where is the division happening in the collective motion of epithelial cells. So there is studies on that. And what people have shown is that if you have a wound, the front few rows are spending their energy mostly into the migration. And then of course there will be gap. So it's not an active gap that is generated. It's just that the cells will become looser, or they will become more spreaded out, and they are just more migratory in nature, and there's space for them to divide. So cells at the back are spending their energy more into the division, and then the front are spending their more energy into the migration. So that's what it's happening.
So is it like the density of the cells at some particular intermediate time will also be less?
Yes. But I think I don't know if this has been explored in a very much detail of how that will impact on the forces, etc. That's for yoga can't.
Okay, and one more thing, like as we are talking about the leader cells, is it like the front row makes those leader cells?
Yeah, so this is also a very good question. There is evidence that also in the back there is cryptic lamellipodia, but we don't know whether it's actually a leader, because a leader means that it has to pull a certain number of followers. So actually, if you look at this study, we actually define the leaders in a very nice way, where you also see the number of followers that are going to, how is a leader defined? A leader is the cell that is exerting a pulling force and is actually at the tip of an outgrowth, and that outgrowth has a certain number of cells. And then when that number increases, there will be a need of another leader. So that's why we only define leaders at the edge. There is also cryptic lamellipodia in the back, So cryptic lamellipodia is actually, this protrusion that the cell is forming. So if, because in the back there's no space, so there's also cell, cells are everywhere. There's neighbors around every cell. So the cell is forming a lamellipodia underneath in a cell that is present in the front. So that is a cryptic lamellipodia forming in the back. Now, if whether that is a leader or not, that part is not really understood clearly.
So am I right in understanding that what is the physical phenotype look like for a leader formation? Is that the protrusion formation?
Yeah. So basically the cell that is a leader is going to be highly polarized, first of all, because there will be actin-based protrusions. You know what actin is? Yeah. Have you heard of actin? So actin-based protrusions at the front of the cell. And at the back, there will be rho A, basically a protein that will enhance the contractility of the cell at the back. So the cell will look like a little bit like this. It's a very large cell with a very long, big lamellipodia protrusions at the front, and nuclei is going to be a little bit in the back of the body of the cell.
Okay, so before you pull your PDMS channel back, there is still the layer of cells is still migrating forward, right?
Yes.
Either through proliferation or migration.
Yeah. So cells are migrating forward. Here.
Right. So what then is the need for a leader cell? Can it not continue to do that without leader cells?
Exactly. So there is actually already an existing leader here. This is the leader cell. There's also another leader here. But what we are trying to show is that a pull from the back is sufficient to create polarization at the front to aid in the protrusion formation, because we are trying to prove that hypothesis that when there is a stochasticity in the monolayer, and there's a wound, that higher forces in the back, in the bulk, is going to aid into the protrusion formation at the edge. So we are just trying to prove this hypothesis by doing this experiment, and then of course, because of the system dynamics of the system, these protrusions are going to die off because there's already existing leaders at the edge.
Maybe a follow-up question on that. So I always thought, maybe I'm wrong, that when you scratch the assay, you were removing the balancing force in one side of the cells which you have scratched, right? So at that point, they need to adhere more and expand more in order to pull the layer or balance the force. So that sets the leadership of the cell, or is that fair to say that because of their being pulled from the back from the other cells, and they need to crawl up more and then make more adhesion points to balance the force?
So that's a good hypothesis as well. The thing is, we have done both sorts of experiments. One where we do not create a scratch, but just confine the cells in a confined EBD chambers, where you actually just create a wall in front of them and then remove the wall, and the cells will actually start to migrate out. Similar to this, I will show you. Sorry, I'm just going to go back to this first. I'm just answering your question. This kind of experiment. So where you just, there's no scratch. Basically, you just confine them and then release the confinement, but you still see leader cell formations at the same length scales. And in fact, this experiment where we did the stress microscopy, where we actually measured the forces behind the leaders, that is in this kind of assay. That's not a scratch wound. And there's a lot more to it. So there's a study that we have not put here that was published in Science when I was doing my postdoc. That's a part of my postdoc work. That I have not put here because that is more into molecular biology. But there we show the role of P53 as well in the leader cell formation, because that is where we did a scratch assay. And when you scratch, the cells that are damaged will have elevated levels of P53. Those will not be able to divide because they are, what do you call them, when there's binucleated, trinucleated cells, the division is impaired. And those cells are actually the ones that will form the leaders. So there's different, molecular and biophysical pathways of leader cell formation. How do they crosstalk is not really clear, actually, to me also.
Because like here in the IBD chambers, they're clearly not getting this death signals, right? They're
so there's no dead cells here, exactly. This is a very clean experiment. So this is the science paper that I'm talking to. I'll show you later on. There we actually do it on we don't do scratch. We don't do this confinement release. That is where we saw that with the scratch, the cells that that are damaged because of the scratch. So that means that that will replicate more of a wounded situation that we have in our body. And that scratch, that damage leads to elevated levels of p53 with NP21 and CDK, all those, that pathway. And that is going to then allow these cells to behave as leaders because they cannot divide. They're spending their energy more into migration. And then eventually at the end of the gap, when the gap is closed, those cells that are unfit are being removed by the neighboring cells. So it's a combination of competition and cooperation. So that study I've not put here actually, but I will show you later on. Yeah, so I'm going to go back a few slides because this part, I how much time I have? I think I've not even covered 1/4 of it. One more, are you guys, whenever you get bored, you tell me, we stop. Don't force yourself to listen to me. Okay, so I was just showing you this part. So basically, when you have this kind of system and you create a scratch, now this is just a representation. You're going to get leader cells here, where you have a higher accumulation of forces. And one can also compute something called a force correlation length, where you can compute how much to what distance in space the forces are correlated. And this is approximately 180, 120, that range for the epithelial cells, which is tens of cells that cooperate with the neighbors. And this is also very well matching with the distance between the two leaders in space. So that means that the leader cell formation, so basically the forces that the collective monolayer or the tissue is exerting not only determines the formation, but also determines the regulation of leader cells at the edge. So in space. You had a question?
Yeah, so I have a couple of questions. So number one is that because the cells are discrete entities sort of connected to each other through some topological network, essentially, right?
The individual cells, you mean?
So yeah, the cells have a fixed neighbor, right? So they are,
Yes,
the neighbor
yes.
is not changing. So my
Not so much,
question
yes.
first is that, isn't it better to sort of plot the correlation as a function of, say, the neighborhood distance, like this is my first neighbor, second neighbor, third neighbor, like that? And the second question is, this is kind of an anisotropic, the forces are distributed anisotropically. So don't you think that having an anisotropic correlation length where you are measuring the X correlation separately than the Y correlation, wouldn't that give you some information?
That's true. So to answer your first question, I think that is how we are doing it. So basically, we are calculating correlation of one cell with its neighbor. So how to what distance the neighbors are correlated. It is correlation length in the direction of migration. So you are taking yeah. And so actually at that time when we were trying to do this study and trying to publish it, the question that the reviewer asked was a very valid question because of these studies that were appearing in, that was actually back in 2014 when I was doing my PhD in Max Planck Institute. So many of the studies were showing that when you actually bias the interface to a more pointed one, you will actually bias the leader cell formation at that region. So if you create micro pattern epithelial cells in a specific pattern where you bias something like this, you make a pointed region, that's where you will form leader cell formation because they show that you're creating more leaders at the pointy edges. So the question that the reviewer was asking is whether it's just, are you sure, is it it's a collective dynamics or how does it compete with the interfacial bias? If you have a real wound, you will have an interfacial bias where you don't create a real straight scratch, but there is always a bias where some cells are already existing in the edge and maybe at this curvature where they are more biased to become leaders. So how does that compete with the collective dynamics or collective cooperativity of forces that you see? So we did this micro patterning experiment. Somebody has a question.
How does the leader cell formation changes with the substrate stiffness?
Yeah, that also I have. I think I have it in the slides at some point. So actually I will tell you, but if I don't have it, I will tell you. We did that experiment also. So what we did was, we know the distance between the leaders is determined by the cooperativity of the forces or to what distance the cells can pull each other. And you can actually try to push them to a much shorter distance in terms of creating a micro patterning bias. So we created these micro patterning substrates where you place cells here, and this is PDMS, and then you release the PDMS and let the cells migrate. And these pointy edges are 75 microns, so much closer to each other than the leader cell the distance between the leaders that you see. And when you do this experiment, this is also a self-explanatory movie that I'm going to show you. So this was the one that we biased, but you eventually see that the distance between the two leaders will come back to the unbiased one, which is a very nice demonstration of showing that the interfacial bias is not really playing the role, but it's more of a collective behavior or the bulk dynamics, or the stochasticity that we saw in the beginning of the talk. And you can do this, you can also increase the distance, and again, the leader cell distance will stay the same. Yeah, so essentially you're seeing this, that the collective dynamic heterogeneity in the bulk is essential for tissue function, where you can actually see that collective migration is impacted by the way that the cells exert forces in the bulk. Now you can go back to what I was telling you in the beginning of formation of organs, where the cells will eventually migrate to form an organ and become homeostasis, or achieve a homeostatic monolayer and keep that as a constant homeostatic monolayer for all of the adult life. The question is, why do the cells need it? Now we already saw that there's some kind of implication of this achieving homeostasis at a certain density for leader cell formation. We can also try to change this density. So if we change the cell density to a very low, somebody was asking this in the beginning of the talk when I was saying that I'm going to show this movie later on. But what we did was we just changed cell density. So this is not no drug, no stiffness, no change in the tissue microenvironment. But all we change is the cell density, where cells are still attached to their neighbors. They're not singular. They're still attached to their neighbors, but they don't cooperate as well as they would cooperate in this case. And what you see is a very drastic change of leader-follower dynamics. So here you see that the cells are migrating very nicely in the front, and there's followers that are following the leader, and the wound is closing much more efficiently. But in this case, where the homeostasis is not reached, or your cooperation is not as much as you want it to be, then the cells are not really able to navigate themselves in the space. So they're not able to close the wound as efficiently as here. And this is the plot. So you can see that this number of cells that coordinate forces with their neighbors here is much less than here, where the cells are pulling and cooperating to a much larger distance.
The difference between the two sides here?
So this is just change different cell density. So basically, when we seed the cells on a in a wound healing confinement assay, we just seed a little bit less cells than the right side. So here there's a lot more cells. So basically the homeostasis is achieved here, but here the homeostasis is not yet achieved. So cells still have to divide and achieve homeostasis here in the left side. You will see a lot more cell division in this case. I mean, you can interpret this result in many different ways. You can also say that because there's cell division, a lot of cell division happening just behind the edge, the cells are not able to spend their energy, all of that into migration. So that is also one way to interpret it. But when you compute the force correlation length in both of these cases, you will see that the cells are not able to pull their neighbors to a long distance. In this case, they are able to pull their neighbor up to tens of cells, but in the left case, there a few cells are able to cooperate, but not up to a very long distance. You got it? So what I'm trying to say is, if I'm a cell, I'm going to pull my neighbor. If I'm at this in this state where I'm not crowded with that many neighbors, I'm not able to pull that many neighbors, just because I'm not crowded with many neighbors.
Wounds at two different places?
These are wounds at two different densities. Density of. So you are which background?
Physics.
Physics. So I can explain you in a different way. So you take some particles and you put them together, okay? And you put them in a very not very dense way. So basically, very relaxed way, where the particles are able to exchange their neighbors. They are also, if they are active, they are also able to divide. But you take the same particles which are active, but are not able to divide and are confined by many more neighbors. So that's the difference between the two. Okay. Okay, so the question now again, coming back to biology. So all this was very nice. You see forces that, or physical forces or stochasticity in the way that the cells will pull their neighbors will have an impact on tissue function. But what about the biochemical stochasticity? Because epithelial cells are active matter essentially. So they are biochemically active. They are dividing. They are maintaining their homeostasis. They are, active in the sense of their metabolic needs. Basically, you have media, or when you eat food that you're eating, the cells are going to absorb that food, and there's energy going on. So there's energetics involved in the system. So they are biochemically active. If you see physical stochasticity, that means that you might also see some biochemical stochasticity, because they're both tied to each other in some way. When I'm pulling my neighbor, this force is being transduced to the neighbor through a biochemical signaling. Biologists will appreciate it a lot more. There's a whole field called mechanotransduction. That is, how many of you have heard of mechanotransduction? So still not many people have heard of it, I see here. So I can tell you a little bit about it. So these the epithelial cells, when you look at them from inside, they are looking a little bit like this. That means that they are tied to their neighbors by some kind of adhesions. It's like a glue tying, sticking neighbors together. And these adhesions are active adhesions because they are linking the internal part of the cells with cytoskeleton. Cytoskeleton, is this term sound familiar? People have heard of cytoskeleton. So cytoskeleton is made of actin, microtubule, intermediate filaments, those kind of fibers or polymers that are inside the cell that will help the cells to do this contractility. So basically, if I'm pulling on a cell, the cell will respond to it using its cytoskeleton. It will change its shape using its cytoskeleton. So actin is one of the most important cytoskeleton that will respond to force that the cell will exert on the neighbors. So we saw stochasticity, or in the physical forces, but then we also wanted to see what about the biochemical stochasticity. I use this term too much. And so the first thing that we want to look at is actin, because actin respond to physical forces that the cells exert on their neighbors. And when you look at this, so basically what you do is you do a simple immunostaining experiment. You take epithelial cells and you stain them with actin. And you can see already that there is cells that have higher levels of expression of this protein as compared to the other cells which have a lower level of expression of these proteins. And how do you make sense of it? How do you put them in the context of how much, how is this protein levels clustered in space, whether they are, similar protein levels with, neighbors exerting similar, expressing similar levels of proteins or different. So, all those questions that you can ask now. So I'm just going to get some water first. It's been an hour almost. Yeah. So to make sense of the spatial correlation of this actin distribution in the monolayer, one can actually go back to, so first of all, what we did was we segmented these cells using cell post tissue analyzer, and we can actually map out the intensity of each cell or actin intensity of each cell. So now you can still say a little bit more in the sense that these cells are having similar levels of protein expression, but not with certainty. So with certainty, you can say it when you actually quantify it using some kind of tools. So we took help of this tool that is used mostly in geography. It's called Moran's index. People have heard of it, Moran's index? No. So basically, this is in short, it's basically telling you, it's a measure of how clustered the entity in space is. So what you can calculate is how much. So if you calculate the global mean of this, all of these cells. So basically, you take cells as individual particles, and you calculate in every particle what is the intensity, and you calculate the global mean, and then you calculate how is it different for individual particles, how is that different from the global mean. And then you can come up with this index where you can plot on the y-axis the spatial lagged intensity, and on the x-axis the intensity of a particular cell. So that means, so what I'm trying to do is, if I'm taking this cell, I'm looking at all of its neighbors. So neighbors you can define by taking the edge of the cells and how many cells touch that neighbors. And one can actually quantify then how are neighbors different from each other. So, if I take this cell in the neighbors, whether how is this cell different from the global mean, and how are the neighbors different from the global mean and that particular cell. So then you can plot this. So the spatial lagged intensity is the intensity of the neighbors, and this is the intensity of a specific cell. And this is following a nice upward curve. That means that the cells are spatially clustered together. Yeah.
WIJs?
So the W is the weight of the neighbors. The small Wij is, I think, the weight of individual, the weight of that we assign to each individual neighbors. I think that is one. I would forward that question to Sindhu because she's the expert. Can you give her the mic? Sorry.
Just give equal weights to all of the neighbors. So if there are, yes, the sum is 1.
Thank you, Sindhu. Yeah, so then you can see that the cells are clustered together in the intensity in the space. And then you can also calculate a local Moran's index, which will tell you, it's an indicator of essentially the spatial association of the neighbors. So basically how a neighbor is different from, so if I'm a cell, how my neighbor is different from my intensity. And then you can plot this nice, spatial correlation plot where you can actually see regions of high and low expressing cells. So what does that mean is that the cells are essentially clustered together in their protein expression levels, and there's hardly these high-low or low-high regions that we see. So meaning that there's no cell that is different from its neighbors, or not so many. So that means that if I'm expressing this protein in a higher amount, my neighbor is more likely to express it also in a higher amount, and so on. So basically, a spatial correlation spatial correlation is what we are looking at. Similar to what we saw in physical forces, where we saw a spatial cooperativity and a spatial correlation up to a certain distance, up to 120, 180 microns, so about tens of cells. Here also, if we calculate this correlation length, we see the similar length scale of correlation in actin. So that means that the cells are clustered together in the levels of, yeah.
I just have a doubt on the online participant. Here, the density is fixed again, no? You have not done
density,
correlation
so we try to do everything at, sorry, I should let you complete first. Can you go
yeah,
ahead?
if the density is changing and then if you find this correlation length, like have you done that sort of measurement?
That's a good question. We have not done it yet. Actually, we are trying to do everything at homeostasis because I'll show you a time lapse. So the next slide that's coming up is looking at this spatial correlation of proteins over time, because in the beginning I opened the talk with the temporal movie where you saw things moving in time. So we want to see how things move in time. Of course, also how these protein patterns are clustered. We see this correlation length. How does that change over time? Does it not change over time, or does it change over time? So in that space, you can also look at the lower density where cells are not really at homeostasis. We have some hints to it, but not really exactly sure, because this is an ongoing work, by the way. So we're currently still lots of gaps that we have here in this work, and we're still trying to explore, understand a lot of it. But I'm going to show you this. Just hold on to your question for one more minute. So first of all, I can tell you that we have not done the spatial correlation for low density samples, but I'm going to show you this plot, and here you might appreciate that at the low density, so cells are basically the actin levels we see an increase in actin levels. And this is because the cells are still dividing, and so the actin levels will go high because they're going through this cell cycle where they're going through G phase and S phase. So the synthesis of proteins is happening, so actin levels are overall increasing. Many cells are dividing, so we see an overall increase in actin levels. We have not done the spatial correlation length for the low density samples. This is the very interesting data that we have that we don't understand completely yet. Those of you who work on biological oscillators might be able to give us some hints on this data that we don't understand. So what we initially thought was, if you look at it over time at homeostasis, the levels of actin will remain constant because the cells are not dividing, nothing is happening. Sorry.
Happening in immune cells also.
Immune cells?
Yeah, and the hypothesis is that this is happening because of this phenomena called actin waves.
Actin waves, okay. Nice.
Yeah, maybe we can talk about
Interesting.
it later.
Yeah. Thank you. Yeah, that would be very nice. So actually, we see this, we don't understand it at all. We are trying to think about circadian clock or some kind of biological oscillators, but we don't know what it could be. What you're saying sounds.
He's analyzing the details.
Who's Sam? Sam. Okay, nice. I'll talk to you later about this. Yeah. So in space, we see these oscillations at the homeostasis. In the homeostasis range where the mean actin is oscillating over time. It's not as constant as we thought it would be. The period is approximately 12 hours is what we see. So that's why we thought of circadian clock, but we have not really confirmed it or done anything about it yet. The other thing that we actually, because this is a mean actin. So this is the actin of the overall population, the mean of the overall population. And then we came across, because then we started looking at biological oscillators. So we came across this nice tool that might be of interest to many of you as well, if you're looking at oscillation or oscillative patterns of any sort, any kind. That's why I put it here. This is also an ongoing analysis, so we are not entirely sure of it yet, but I'm going to put it here because this is cool and it's a nice tool to actually talk about. So this is something called PyBot, which is developed by these people. Actually, Gregor Monke was the person who developed it in his PhD, I think. And this is a very nice tool that you don't need programming language to use this tool. This is an online tool that you can download and you can actually put your data and look at how things oscillate in space. You can compute the most dominant frequency. So what we did here is that this is the most dominant frequency of actin. So you take a space of cells, actin intensity, and then you divide them into smaller grids. And you look at in each grid, so this grid is the size of a cell. And in each grid, how does things change over time? And then in every grid you see that, and then you compute out the most dominant frequency. That will actually give you the signal that is mostly dominant in the whole space. And so this is what we saw, an oscillation or oscillatory signal, and we see this repeatedly in all of our experiments, which we don't understand it completely, but we are hoping to in the future. Not only in actin, but we also see this similar, actually, sorry, one more thing that I wanted to bring here is if somebody wants to know whether that is somehow impacting on the correlation length of actin in space. So we also saw that at the peak, if you look at the images at the peak versus at the valley, you have a much higher correlation, or not much higher, but a little bit higher correlation of actin at the peak as compared to at the valley. So that means that the cells are clustered or cooperative. They have a higher levels of cooperativity at the peak than at the valley. What does it mean for the tissue function, for the wound healing, for cancer initiation, for nutrient absorption? We don't know, we don't understand it. So that is something that's ongoing in our lab right now. And again, so a similar kind of analysis we could do for traction forces, and again, we see the same. So this is, I think then you were saying the actin waves, it could also be linked to the traction in some way, because the traction forces are essentially tied to actin. Yeah.
One thing that I must comment on is the time scale. So when we are looking at the immune cells, so the time scales are of the order of a few minutes, not more than that.
Oh, okay.
But here it is.
This is hours.
A few hours, but it is not, say, the period is not, say, 12 hours. There are some shorter oscillations. That's also.
True. Yeah.
So there can be multiple factors that are involved over here.
That's true. And it's actually, it's difficult to dissect because these cells are also not synchronized in their cell cycle. So these cells are placed in a dish, and they may be at a different point in their cell cycle, and that might also be contributing to many. There's actually oscillations in the oscillations. There's many factors that are playing their role, and we need to basically what you call it, detrend things and look at individual entities on their own. And that's what we're trying to do. We're going to synchronize the cell cycle, and then we're going to look at the circadian clock separately. So we are going to dissect it slowly over time and see what what it means. And then, of course, we want to see what it means for the tissue as a function. How does it change the function of the tissue? If you, for example, if it's linked to the clock, if as you get older, you're getting more diseases. That could be related to something like that. But this is all speculations and we don't know yet.
So Medhavi,
Yes.
technical question. So how are you doing the circadian live reporting?
We have a live reporter now. We are going
You
to
have a
do
live
it.
reporter that is in MDCK, or is it?
So,
I thought like the only one is in 3D3s.
Sorry.
I thought like the only available one on the community is in 3D3 that.
No.
Zavitra Path was.
Yeah, that one is there. I
Yeah.
think we got the same plasmid. It's a lentiviral plasmid, and we can actually make our own cell line with the live.
Okay, maybe I can come and talk to you.
Sure,
Because Shaun and
yeah.
I tried to do some 3D structures using that.
Yeah,
Didn't
sure.
work.
Sindhu, can you comment on it? The plasmid that we have, it's a lentiviral plasmid that can be transfected to mammalian cells also. No? Xavier,
Yeah.
Tapar.
We are trying to use the same plasmid to make
So
stable
it's the
cell line.
PMOL, right?
It's the PER2 gene.
PER2 gene. Okay. Okay. And with the, so I think they're also looking in the sensory pathway along with that, right?
Yes, they were also looking at YAPTAs and how that affects
Yeah.
the circadian
Okay.
periods.
Great. Cool. Thanks.
Yeah, thank you.
I'll
We'll talk
come
about
and
it.
talk to
Sure.
you.
Yeah. So this is all ongoing work and work in progress. Things we don't understand, but that's the fun part of it, I guess. The final part of after this, I have just few more slides. Just bear with me for a few more minutes. One can also think of how does the biochemical stochasticity crosstalk with physical stochasticity, and we can do this, use these tools that are developed in the field by physicists. It started with, I think, a cell fit, that is a tool to infer forces from the cell shapes. And one can adapt that tool to actually calculate the pressures and the line tensions of individual cells with their neighbors. And what we saw is that the pressure is negatively correlated with actin intensity. So the cells that are having a higher actin levels will have a lower pressure. So there's some kind of crosstalk between the biochemical and physical stochasticity that we see, but we again don't understand it completely, and we are still working on it. If you have any ideas, welcome. Give us the ideas that we can work on. Okay, so this is all that I already talked about. There's stochasticity that has relevance to tissue function. I showed you in terms of wound healing and leader cell formation. And then there's biochemical stochasticity in space that reveals clustering of cells of similar protein expression patterns. There's oscillations that we see in time in both actin and traction forces that may be linked to tissue function, but it's yet to be explored. The last few slides I want to show you is completely different. It's a little bit more biology project that we are working on. So these few slides are more for biologists in the crowd. How many are biologists in the crowd? One. Let's see if you have any questions, others please ask. So basically, this is a problem of cancer initiation. When you have I don't want to say when you have a cancer, that's a very bad statement to say. Okay, so if a tissue is mutated with an oncogenic mutation, that oncogenic mutation is slowly going to expand. So that oncogenic mutant is slowly going to expand in space. There's a concept called field cancerization, meaning that groups of cancer cells are clustered together, and that group is going to grow bigger and bigger and then form a tumor eventually. There's an oncogenic mutation, and then there's other cells that will become cancerous, and this will go on and spread in the tissue and then eventually will form a tumor. So the question that one can ask is where's the point of no going back? So if you have a smaller cell versus if you have a bigger group, when is the point where you cannot evade out these mutants from the tissue? And this is coming from the fact that there's recent studies that show that if there is an unfit cell or an oncogenic mutant in the tissue, that is going to be extruded out or recognized by the neighboring cells and be extruded out from the tissue. But one can ask where is the how big cancer mutants should be where they cannot then evade extrusion.
The question I was going to ask, like when you say how they can be eliminated, are you talking about the immune cells eliminating the cells,
So,
or is it
yeah.
like the force they're getting
No, so-
extruded out of the tissue?
Yeah, so no, I'm not talking about the immune system here because the immune system is not yet in place. So actually, this is I have not put it in a very much detail. So this is another field that we are working on in the lab, and don't present it to this audience so much because we are more of a physicist here. But I'm talking about the competition between the neighbors. So meaning that if there's epithelial tissues, if in the tissue, one cell will attain an oncogenic mutation. How does that compete with its neighbor for survival? So it's a more evolutionary approach of its immune system will come into picture a little bit later, I think, when the oncogenic cells are a little bit more or not. I don't know yet actually. So at the initiation stage, whether the immune system is involved or not is not completely known.
So
in the field.
the question that comes to my mind, maybe it's a bit naive, but if there is a force imbalance between the neighboring tissues and the cancer cell either is going to compress the tissue or it's going to get out of the tissue. So, but either of those processes are actually bad for the tissue itself. I mean, so when you are saying that, what is the point of going back? Isn't that so? How this mechanistically you stop that process? Like if I'm saying like I need to have enough numbers when the tumor so-called is going to press, or the tumor is going to get pushed out and form a bulge and then get out of that. So what do you mean? Like at some point there will be nutrient competition that will outperform the tissue, will outperform the cells, or there is like other measures that gonna come and degrade the cell? And
So actually there's many components of this problem. One of the components is the health of the tissue itself, because when you talk about competition, you talk about fit and unfit, you talk about winner or loser. And the winner can be, there's a whole cell competition field that talks about it a lot. And actually, it's been shown in many of the papers that some of the mechanisms are also known actually. How do the cells compete? How do the tumor attain this super competitor status where, it depends on the mutation also. So, the one mutation or it's an accumulation of mutation. There's many aspects to it. One can try to dissect and understand it one by one. So, of course, tissue health, the group of the oncogenic mutants, the size of the mutants, and then the mutation itself, what mutation the oncogenic cell is harboring, combination of all of this, and of course then the immune system. So many things actually are in place, and many people are actually looking at many of these aspects. But it is well known that cell competition is an important part of tumor initiation. Now, at least in the last 10 years, it's been a lot more, there's been a lot more work on it. That it's not only the tissue microenvironment or the immune cells and the mutation itself that is playing a role, but it's also an active cell-cell interaction that is playing a role in determining whether the tumor will stay or not. And how does that active cell-cell interaction play out? What are the mechanisms? All of that is not really completely understood. Some mechanisms are known, and there's a lot. Some papers talking about the involvement of cytoskeleton, filament, actin, which is a very intuitive thing to think about. When the cells are, because they're mechanically active cells, they might also involve the cytoskeleton to eliminate the unfit cell. So that is known, but a lot of mechanisms are not known. And actually, one can use these, if you understand it, we can use it in therapy, because we can fortify the defense mechanism of host cells and then allow the tumor to stay put instead of forming. So that's what we are trying to do. We're trying to understand the initiation of cancer. And so far, what we have some data on it. Now, these movies are not of the same contrast, but I hope you can see them clearly. So this is one mutant that you will see getting extruded. But here, do you see many?
Sorry, technical question. Are these monolayers or?
These are monolayers. So we are also developing organoids now.
Okay.
We have also already developed it. I've not put it because we have not introduced mutation in the organoids yet. But we are working on developing organoids of various sorts of breast.
What suspension-based organoids or what types?
Different types of organoids. So we have breast organoids, which are acini structures embedded in collagen gels. So those are suspended, but not in media, in hydrogel. And then we have oral mucosa organoid that we are currently working on to develop. That is an airlift organoid, which is stratified tissue in space. And we are going to actually infect them with lentiviral vectors, delivering the oncogene, and we'll see how the cancer will initiate. So good that the movies have stopped. I can comment on it just a little bit and then finish in the next slide. So one can actually make an inducible construct of lentiviral vectors, which will deliver the oncogene to these monolayers. And in this case, there was just one oncogenic mutant that was extruded. But in this case, you see groups of oncogenic mutants that evade extrusion, that do not get extruded. And how does this competition between a single versus a group play out? And how do they evade extrusion when they are present in a group is not really understood. So that's what we are trying to understand right now. What we see here is a very nice, some kind of separation between the normal cells and the oncogenic mutant. So that's what we saw when we did this microscopy. We saw a very nice separation of these cells in around egg-like structures. And we tried to stain them with actin and see what that means. And we saw this nice belt of actin. So there's a contractile belt of actin that will prevent the oncogenic cells from getting evaded from the tissue. And now this is a very nice observation, which again, we don't understand completely, but these contractile actin belts are not seen very often in the homeostasis monolayer. So these are just belts that allow the cells to achieve some kind of cooperativity or do some kind of activity. If there's a movement, you will see a contractile belt. But why do you see a contractile belt in the middle of the tissue is not really understood. And it is a part of the group mutation is playing a role here.
But this contractile actin belts are observed if you do like some sort of this is the pars string kind of wound healing process.
Yeah, so
right?
Movement it's seen. So
Right.
To achieve some kind of cooperativity that is seen. But in this case where you have a homeostatic monolayer, why do you see this contractile actin belt around the tumor cells?
That comes from you have force imbalance. Like in wound, it will come because cells have the cells at the periphery, they have forces in one direction, not in the other direction. And they will, and they are not adhering, and they will form this belt. So if they're not, there is a adhesion imbalance between this tumor and they should form a. Is it something
for you it is intuitive?
I think.
Okay, nice. I'll talk to you about it because I don't know, we don't understand it. Why the cells are
maybe the adhesion between the cancer cells and the regular tissue cells are going to be different. So if I think of this as a sort of like an interfacial tension between those cell lines, that is different from the others.
Yeah.
And they will deposit.
It could be related to differential adhesions. It could be related to, we are trying to do an experiment where we are, because why we're doing this experiment is because of this result that we saw. So we did this, we tried to plot this velocity correlation length, and we saw a much higher movement correlation in this case versus in the case where you don't see the belt. So we are thinking that it could be related to invasion, where the tumor cells are not getting extruded and they might be preparing themselves for invasion into a matrix. So the part that they don't get extruded is also an interesting fact. I mean, if they are single or they are 2-3 cells, they will get extruded. But if they are in a group, they will evade extrusion by forming this belt. Now, this could be just a differential adhesion, like you were saying that, there's just a belt forming because the cells are different. These are HRAS mutants and these are normal cells, and they adhere differently. So they will form a belt.
Okay.
But it could also be related to some kind of cooperation that is happening inside. And that idea we got from a developmental biology paper where people were showing this actin belt from forming in the case of blastocyst when cells were trying to migrate out. So we don't know yet. We have to do some experiments, and I'll talk to you about the adhesion part also. We can maybe also stain for some adhesion molecules and see.
Yeah, and these are like on monolayers still, right?
This
so
on
they're
monolayers.
Still on monolayers.
Yes.
Okay.
We are going to put it in an organoid setting and to see that, but first understand it in 2D and then go to 3D.
And these are all MDCKs, right? Still
no, these are MCF10A, so these are breast epithelial cells.
That's beautiful.
Because we wanted to do breast, we actually made MCF10A acina structures organoids in the lab. So we're now going to do the same experiment on the 3D organoids. This is MCF10A.
Okay, maybe I can talk to you. We have some other similar things, but different. Sure. Yeah, we can talk.
Sorry, I did not mention the cell line. This is a breast epithelial cell. So we're looking at breast cancer initiation. That's why it's coming from. Okay, sorry. This is how the students gave it to me. I might be. Who gave me this data? Tanya, do you know about this scale bar? It has to be in micron. Sorry. That's how it was given to me. I can only do so much, you know.
I think
This is not right. Yeah, we have to change it. Sorry. So this, yeah, cell size is approximately 20 micron. Yeah, no, this is wrong. So it has to be, so this much will be approximately 50 microns, I think 50, 60 in that range. Okay, any more questions before I finish? Yes.
Oh, I just had one question about in the beginning slide, you showed the stochastic like stress landscape sort of
go
thing,
back
right?
to that slide? Or
No.
Okay.
I was just wondering, like, how are you saying this force landscape is like stochastic? Is there no like way to quantify like this from like something like a cell boundary, like from something I'm not sure, but like something like a micro from the micro scale? How can we get like the force? It's just like, how are we saying this boundary is stochastic? Is there no deterministic?
That I showed in the
Is there no deterministic method behind
So
that?
we have actually in the paper that is cited there we have quantified everything. There's also levels of forces that we show that are different in different regions of peaks and valleys. And of course, it also changes in time. That's why it is a dynamic heterogeneity, its property, because it's dynamic in time and it's heterogeneity. It's heterogeneous also. So in space and time, it is stochastic. That's why I called it a stochastic landscape of stresses.
But like, then what is like the main source of like the like in some regions you have lower stress, in some regions you have higher stress. So what is like the main like microscopic source of those?
Yeah, so the microscopic source of it is the contractility of the cells itself, because the cells are biologically active. They are exerting. So we calculate it from the cell traction. I mean the cell force that the cell exert on the substrate. So we are calculating the displacement of beads underneath the cells, and from that displacement we are calculating the force, how much force you need to cause that displacement. So it is a force that the individual cells are exerting on the substrate, and it's a property of the contractility of the cell. And so the source will be the, biologically active cells that are different, that have differential contractility in space. And that is why we then brought about the actin landscape in the picture, because we wanted to see how it crosstalks with actin. You got it? More questions?
You showed a plot showing the valleys and peaks. So what are those valleys and peaks exactly?
Sorry, can you
there was a plot where you saw the valleys and peaks. So
which plot?
This one, I think.
This one, okay. So here the peaks are images at this, and the valleys images at this. So we are looking at the spatial correlation at the peak. There's oscillations in actin levels, and the levels, the space where or the images where we see higher levels of actin, we took those images out and we computed in space how cells are correlated, and that is the peak and the valley.
So this is not a time correlation, a space correlation.
This is a space correlation. Okay, if there's no more questions, I go to the most important slide. Where is it? Here. So I want to thank all of the people actually who've been working on this project very, very hard. Sindhu is the one that's leading the Stochasticity project. She's sitting right here. She's an expert. So all of the questions after this, direct to her. And Tana is the one here who actually joined later than this picture was taken. So therefore, she's edited here. And she's the one who did the PyBot, the oscillations analysis. And so both of them are working on this project very actively right now. If you want to have any discussions with them, please go ahead with that after this. And the funding contributions that, the agencies that fund us here. And I thank you all for listening. Where is my laptop? Where's my laptop? What's in there? Okay.
Between two patches of leaders, that is roughly 200 micrometer. So what controls this distance? What sort of?
So yeah, so actually this is a very good question. In the paper you will find, if you go back to the slide, there's a citation there. In that we try to understand a lot more. We did actually a modeling and a simulation. We collaborated with physicists to understand this. And I can tell you a little bit briefly. So a leader cell is exerting a force because it's pulling. So if you put a point force on the leader, that point force will have a certain decay length. So that decay length will determine the next formation of the next leader. If the decay is faster, then you will get leaders closer to each other. If the decay is slower, and the point force can be transduced to a longer distance, then you will have a leader at a longer distance. So longer length scale. And we actually tested it using experiments. So we tried using drugs which will enhance or reduce the contractility of the cells. And if you increase the contractility, that means that you're increasing the length up to which the cells can pull. So then the distance between the leaders went really high. And if you reduce it, then your distance between the leaders are reduced. So the source is again the contractility and the ability of the cells to pull their neighbors.
So is this related to the stress correlation length?
Yeah, so they are both same. So basically the amount of forces that the cells can pull, basically the correlation of the stresses in the bulk is exactly the same as the distance between the leaders. So they're both, it's essentially the property of essentially the property of the tissue, of the individual entities of the tissue, which are the epithelial cells. And if you change the cell type, then you will change the distance and everything else, because that cell type will have a different ability to pull.
And the second question is about basically, do you know if people study these things using vertex model?
Yeah, there is a person that is working with us now is in IIT Bombay. They're working on it, and we are trying to develop a vertex model to understand the molecular mechanisms of it as well. There's other people like Pascal Silberzahn. Do you know Institute Curie? I think he has done some of this work on using some theoretical models to study selective cell migration on epithelial cells. Then I don't remember other names, but there's definitely people to look at selective migration in epithelial cells.
Yeah, I asked this because this is probably not a very straightforward.
Yeah. It's a very convoluted system because it is soft, it is active, and there's a lot of signaling involved as well as the forces. So there's a crosstalk between biochemical and physical pathways, and so one has to do some modeling as well to understand it completely. More questions? Yes, go ahead, please.
So I have a question about the first part of the talk, where you showed the stress profile of the whole layer. So I have heard some terms like positive and negative pressure wound healing. So can you explain that term in context of those stress profiles of a tissue?
Can you explain the positive and negative pressures first, where you
so
hold it? Because then I will be able to get the context of the question.
So essentially, when a wound is healing, so it is working against the air pressure, right?
The air pressure. So I don't think the air pressure will have that much of a role to play here, because if you think about our body, this will not have a difference in air pressure that much. So if you mean the imbalance of the forces because there is a gap, certainly, do you mean that? I mean, if you talk about pressures, I can understand that the pressure that the cell is exerting at the edge, experiencing at the edge will be different than the cells that are experiencing in the back, because the edge cells are not surrounded by all neighbors. The edge cells, one side of them is free, and the behind, they are surrounded by neighbors. So, there's a imbalance in terms of the pressures that they might be experiencing. So it could be related to that. Is your question related to that?
No, actually, when we have a, let's say, tissue, and let's say we scratch it, and so the growth, the expansion happens in the opposite direction to the scratch. There are some type of tissues with this dynamics, and some type of tissues when.
So you're saying that you scratch it and the expansion, but how would the expansion happen? The opposite direction, there's cells everywhere, no? So there's the gap is only at the place where you scratched. So how can the expansion happen in the opposite direction?
So,
I don't understand.
So let's say we have a, yeah.
Okay. Because maybe I don't follow your question.
Okay.
So after this, come and talk to me. We can then understand each other.
Okay, thank you.
Any more questions? Yeah.
I don't know if this has been answered, but like I didn't get. Like, why are leaders actually required in the process of wound healing? Like, if let's say we don't have leaders, or
let me think about your question a little bit. So why are leaders required? I think they are required because the cells will need to know which way to migrate. Actually, there's many aspects to answer your question. One is in terms of chemotaxis. If you heard of chemotaxis, have you heard of chemotaxis? So basically, in migration, in general in migration, there will be an individual that will sense the nutrients. So in chemotaxis, where you will see the gradient of the nutrients coming, and this individual is sensing the gradient and is migrating, and the others are, and because this is migrating, the others are going to follow it because it's tied to the neighbors. So it's when it moves, it will pull, and then automatically this leader follower behavior will be established. It is like a flock of birds that you see in the sky, where you see a V-shaped structures, and you see a leader follow behavior there. So we see leader follow behavior not only in cells, but in many other aspects of nature, in humans, for example. So I mean, in birds and in humans, it's a different story, because there's a conscious choices that are coming into play. But in cells that are all essentially the same, the need of the leader will be to actually allow the collective motion to happen, where, which way the movement is going to take place. So one cell is moving, and the others are following because of the pull from this particular cell. In the neural crest cells, for example, and in many other cell lines that are not tied to their neighbors, you will see leaders exchanging. In development, you will see those papers also exist that, during development, when the organs are forming, when the cells are going from the, embryo to migrating towards the organ site and forming the organ, those cells are not epithelial-like. If they're not epithelial-like, they will be migrating. One will be the leader, because so that their migration is happening mainly of chemotaxis. And there the leaders are exchanging a lot. So there the leader is not fixed. But here it's mostly because physical contacts of the cells that the leader-follower behavior is very strongly established. That was a very long answer, but I hope you got the context of it. Did you get it? Yeah. We can talk later also. Thank you.